Okay, it's 3 minutes after the hour, so let's get started. Hello, everyone. Welcome to week three. Week three was about gene curation, and I will get us started on the live Q&A. So for our agenda, let's start with admin updates. So next week is the last workshop. We'll be discussing actionability. So this week in my email, I will send out a quiz with questions related to all 4 weeks. This will not be a lengthy quiz, likely one question per week, if not maybe an additional question or two. And completion of this quiz, but not the overall score of the quiz, I'm not going to grade the quizzes, is required to mark your participation in this workshop. We just want to make sure that everyone is paying attention to the emails and participating in a way that makes sense. And I'll reiterate this via email because I know that some people have been sick or have had internet issues. But I just wanted to mention that. Expect the quiz in the email this week. So now, like we have been doing for the last 2 weeks, we'll tackle questions from the forum first and then live questions if time. I anticipate we'll have time for those. So, and they'll be grouped by theme again. So the first one was a great question about video accessibility. So in the spirit of diversity, equity, and inclusion, has ClinGen considered incorporating sign language interpretation or other accessibility features into the videos shared on social media? So this is great feedback, and I'll bring it back to our team. In general, we recognize that accessibility is very important, but because we are NIH funded, we don't have a ton of funding to be able to add either translation of materials or accessibility features to our content. But we could explore doing some of this in at least a subset of like really critical videos, like, one video about ClinGen, an overview of it and some of the curation videos. So I'm going to check in with the team and see if this is something we could potentially budget for in the future. I know that there are some closed captioning tools, but they tend to fail pretty badly, at least from what I've seen at scientific meetings when it comes to a lot of scientific jargon. So we'll work on that and explore that, but it's great feedback. Okay, so a bunch of the questions now were related to the gene curation framework itself. So one was, what is the main goal of the ClinGen framework and how does it accomplish this goal? So the main goal of the gene curation framework is to determine if a gene has a legitimate relationship with a disease. And when we're saying diseases in this case, we are considering diseases that are monogenic. We really don't have complex gene curation frameworks at this point, and relatively moderate to high penetrance. I think we're starting to explore some lower penetrance conditions, but the framework is really optimized for monogenic conditions with moderate to high penetrance. So determining if a gene has a legitimate relationship with disease like that, such that testing for this gene would yield diagnoses. It's often used to determine which genes should go on clinical testing panels. And per the ACMG statement that I sent to you, those genes with a strength of moderate or above using the ClinGen framework should be included on diagnostic testing panels. The next question was a question about disease entities. And so we term that in ClinGen lumping and splitting, whether disease entities should be lumped together or split apart. So the question was, how do expert panels handle situations when the same variant contributes to two different mechanistic disease pathways? So that's a great question. And before we perform a formal curation, which was maybe mentioned in the training for gene curation, we evaluate what assertions exist for a gene in the literature or anywhere published or in ontologies, places like OMIM, Orphanet, the Monarch Disease Ontology, as we know that genes can be associated with multiple conditions. So in this analysis, we call it a pre-curation. So the curation before the curation, we determine the phenotypic spectrum, the mode of inheritance, and the molecular mechanism of all of these gene disease assertions. And in general, we need to weigh the similarities and the differences for all of them. So in general, we really weigh heavily differences in modes of inheritance and molecular mechanism. So if there's no difference in variant type across multiple assertions, if the mode of inheritance is the same, we often combine these conditions together into a broader phenotype label, because we feel strongly that molecularly, if you are given a variant in the absence of a patient phenotype, you should be able to tell if that variant type is associated with a particular condition. And so if you can't tell based on the variant type, and I should say, like, caveating missense variants, if you truly knew that the missense variant was causing a loss of function versus just being a missense variant, because we know those are nebulous. If you can't tell based on the variant, we tend to group all of those conditions together into a wider, more broadly named condition. But we do specify in the curation on the ClinGen website, these are all of the different claims that we have considered together when we are evaluating our gene disease relationship. Another framework question: should clinicians act on variants in limited evidence genes when no alternatives exist? So I would say in general, no, because you just don't have a good sense of whether that's a legitimate gene disease relationship. In our experience, many of the limited genes remain limited for years and years and years. And this could be for a variety of reasons. It could be because the disease is rare. It could be because there's not a legitimate relationship between that gene and that disease. It could be that the populations where this is a legitimate cause of disease or a significant cause of disease have not been represented well in the literature or tested, but we're just not sure which one of those it is. So if you have a candidate gene, usually limiteds are even a little low for candidates. Most of the time it's like a high limited, almost moderate. If you have a candidate gene, really clinical research studies are used to generate enough data to bump those genes up the ladder to at least moderate. So cohort studies, animal models, experimental studies, and there's often funding for this at like the government level, at least in the US, to really recruit rare disease patients and try to work them up. And if you find a candidate, to develop an animal model or at least do some functional studies to try to validate that. So per the policy statement from ACMG, only genes with moderate or higher gene disease relationships should be reported in diagnostic testing, and suggested you should go even higher than that for screening panels. So strong and definitive genes for screening panels, because those individuals are unaffected. Clinicians could also consider registering their patients or genetic results of their patients from genetic matchmaker processes to try to assemble a larger cohort to increase gene disease evidence. So there's different nodes of the matchmaker exchange that clinicians can upload the genetic data for their patients and a list of phenotype terms and see if they match any other patients with similar phenotypes that may have variants across different genes. But so in general, you should probably not act on limited evidence genes. Okay, another framework question is, what does it take to become an expert in a specific gene? So if you look at ClinGen's expert panels, they have members with a variety of expertise. So these could be clinicians, researchers, genetic counselors, and laboratory personnel. So in order for a curator to become more specific or more experienced in a specific gene or disease area, and this definitely happens across ClinGen, I encourage a curator to join a disease-specific expert panel to listen to the discussion and curate for them. So I've been involved with ClinGen for the last 10 years, and that really has helped me develop expertise in many disease areas. I sit across 15 different expert panels, and just from participating in those expert panels and listening to the discussion, I have learned so much from those experts. So honestly, that is the best experience that you can have, and we're always happy to have volunteers. So to volunteer for ClinGen, and I mentioned this in the first week, visit clinicalgenome.org/volunteer and take the volunteer survey. We're always in need of curators. And we're always particularly in need of gene curators, because people don't necessarily always understand about gene curation. I think variant curation is very well known. Everyone recognizes that variant curation is really important, but gene curation is just as important, because you need to determine a gene is related to disease before you could even report variants in that gene. Okay, the next one is also related to ClinGen gene curation. If I were to do my first gene curation, what are the most common mistakes made by beginners? Like population frequencies, loss of function, gain of function mechanism, over interpreting a single paper. It's a good question. I think the things that this person listed are some common mistakes that new users of the framework can make. One of the most common, in my opinion, is scoring a variant without checking its frequency in population databases. As I'm sure you're aware, many of the gene curation papers in the literature are some of the older seminal papers, and they really were around when there weren't large population databases. And so it can be quite common that some of those variants that were absent from 100 control chromosomes, for example, are actually quite frequent in Nomad. And so many GSEPs will actually have minor allele frequency cutoffs for their disease area, kind of like how VSEPs have this. Um, and they will look at the frequency in Nomad and say, is this rare enough to be suspicious and we can score it, or is it not? So it's important to check the frequency of variants to be scored in gene curations and making sure they're not too frequent in the population. Another common error or confusion for new curators is scoring loss of function variants at a high score when it's unclear if loss of function is the mechanism of disease. Our framework is particularly weighted towards loss of function variants and loss of function genes getting more points just because it's easier to predict loss of function variants than it is missense. You just don't know what missense are doing oftentimes. But curators should ask themselves the question, is it true or is it suspected that loss of function would actually be a mechanism of disease in this condition? For example, in RASopathy, it's very well known that gain of function is the mechanism of disease in most genes and the MAP kinase pathway. So you wouldn't want to be scoring bona fide predicted nonsense mediated decay variants in that case. So that's another misconception. One additional confusion is scoring multiple family members in the case level data section instead of scoring the proband there, because you should really only be scoring one proband per family in the case level section and scoring the family members in the segregation section. So that can also be a common misconception. One other thing I didn't put here that I just thought of that comes up often is evaluating knockout animal models and the zygosity of the knockout animal models. So you may be familiar with many animal models needing to be biallelic knockout to cause a phenotype. And so it's important to consider, does that match the proposed mechanism of disease in the humans with variants, or is it just a knockout animal? So is it an autosomal dominant condition and this is a biallelic knockout? That might still suggest that the gene is critical and may be related to the disease process, but you might want to downgrade points there. And the expert panels have discussions about this. Or is it a RASopathy gene, for example, and gain of function is the mechanism, and therefore you wouldn't want to have a knockout mouse showing that the disease is recapitulated because that's inconsistent with the mechanism in humans. So that's another potential common misconception. Okay, another question about the framework. How does the ClinGen framework balance genetic evidence, so case level, case control, and experimental evidence, functional studies, model systems, rescue experiments when scoring a gene disease relationship? So when we developed the point-based system in 2017, we decided to give genetic evidence the range of 0 to 12 points and experimental evidence the range of 0 to 6 points. So this was intentional. We built the framework such that genetic evidence alone could get a gene to definitive. You cannot get a gene really anywhere if you're only scoring experimental evidence points. That would still be listed as no known disease relationship, unless there's an animal model, and then it would be animal model only. You need human data to be able to get a gene curation above no known disease relationship. So if a gene has maximum experimental evidence, so 6 points, and only a little bit of genetic evidence, it usually can only get to a high limited or low moderate. So in general, my experts and my expert panels do like to see a balance of information and would be wary of scoring a gene highly, saying it's definitive, for example, with imbalanced data. So most of the time, if we did even get to 12 points with genetic evidence, but there was no experimental evidence, they might still decide to downgrade that curation manually to strong until more evidence is published in the literature experimentally linking that gene to disease. Um, Another thing that we thought about when we set this framework is we capped certain types of experimental evidence by saying that certain evidence categories within experimental evidence can only get 2 points, for example, or 3 points. So, for example, animal models can only get 3 points because we felt that you weren't, at a certain point, you weren't getting additional information by scoring the same types of experiments over and over and over again. And so we built in a little bit of a checks and balance system there, that you couldn't just score expression data, for example, that the gene is expressed in a tissue related to disease 6 times and then max out genetic or experimental evidence, because that wouldn't give you holistic information. So we really did that to encourage scoring a range of experiment types if possible. Okay, another question on the framework. How does ClinGen handle situations where strong functional evidence exists, but human case level evidence remains limited due to underdiagnosis or lack of access to testing in certain populations? So, experts will often think about the rarity and the phenotypic specificity of certain conditions when scoring the limited amount of case level data they have. However, they can't score absence of data, so this can only go so far. So, as mentioned on the previous slide, genes with a bunch of experimental evidence could at least make it to moderate if you had a case or two for genetic evidence, but it likely wouldn't go higher than that if not much genetic data are available. Because if you think about it, the line that expert panels are often thinking of is the limited to moderate line, because that is really telling you, should you add something to a clinical test and put it on a panel, or should you not? And limited genes should not go on panels and moderate genes should. So they're really thinking about that distinction when they're deciding if a gene should score at limited or should score at moderate. Next one's another framework question. So, what's the rationale for not assigning quantitative scores to contradictory evidence during ClinGen gene curation? So, we did consider giving negative points to contradictory evidence in this framework while we were developing it, but we felt that the weight of contradictory evidence pieces was so variable that it couldn't be accurately quantified, and that expert judgment was really critical. So, for example, at the lower end of the spectrum, if you had one single variant that was published and it was maybe only seen once or twice, it's in an early publication that you found to be very frequent in Nomad, and you shouldn't score that one. That's one piece of contradictory evidence. But let's say there were 10 or 12 other variants that were rare enough and suspicious enough to score. That is not that heavy of a weight if that one single variant turned out to be too frequent. However, there is the other end of the spectrum of maybe case control data analysis showed that there was no significant enrichment in cases versus controls. That's a pretty big piece of contradictory data if it's a well-powered study. Or we've had some in the hearing loss domain that all variants previously identified and thought to be causative were actually in a pseudogene. That's highly contradictory, and you'd probably have that as disputed or even refuted if they had published that information. So we really wanted to leave that flexibility in there and kind of require expert judgment a little bit when weighing pieces of contradictory evidence because they varied so much. Another framework question, what are the recommended steps when key literature needed for curation is behind a paywall or otherwise inaccessible? We recognize this can be a difficulty. So in our expert panels, we all have different publication access, as you know, we're at a range of different institutions, and we will often email each other to help with access of relevant papers. I actually just put in a library loan request the other day for articles for a colleague because there were only 3 articles total for this gene disease relationship, and she didn't have access to 2 of them. So we do try to work together to help each other unofficially to break that paywall. Okay, the next question is about curation prioritization. In cases where a gene has strong evidence for one phenotype but weak evidence for another, how does ClinGen prioritize which phenotype to curate first? So this is a great question, and maybe one we didn't necessarily think about in the beginning when we were kind of organically growing ClinGen, because it was kind of like experts would come to us and want to start expert panels, and it wasn't necessarily systematic of we need to start expert panels A, B, and C because a gene is associated with all of those conditions. It was really just these are the experts who are excited. and it's because we're largely a volunteer organization. These are the experts who are excited, who are coming to us to start expert panels. But now that we've really filled out many of our disease areas, we're starting to think about this prioritization. So we're actively exploring how we can do this. So in theory, we would hope to prioritize those stronger evidence assertions first, because you don't just want to come to our knowledge base and see this gene has a limited relationship with disease when it actually has a strong or a definitive relationship for another condition. But it's kind of something we can't help at the moment while we're prioritizing. Um, and it doesn't always happen because different gene curation expert panels may tackle different disease claims for a single gene. You can imagine that there might be an isolated body system condition versus a syndromic condition. For example, I worked a lot with the hearing loss expert panel, and there are some genes associated with hearing loss and then also skin conditions, or hearing loss and then higher non-syndromic hearing loss and then endocrine conditions. Oftentimes when there's a more syndromic condition, the experts in the hearing loss domain didn't feel comfortable tackling those. So we would have to reach out to another group to try to get them to prioritize it. So we're exploring ways to do this in our prioritization system, particularly for those genes that are highly actionable. And we'll talk about actionable genes next week. Those that are suggested to be returned in a secondary findings setting. We really want to make sure those have complete sets of curations. But I would say that just because there is one curation for a gene on the ClinGen website that says limited, that doesn't necessarily mean that's all of the potential curations that would exist for that gene. And I do suggest that you go check out the Gene Curation Coalition database, and that was talked about a little bit in the recording. That is a public repository for gene curations, because we have about 22,000 curations on that website, which is search.thegencc.org, as it might be a bit more complete than what ClinGen has right now. So we have a question about recuration. So if a gene reaches greater than or equal to 12 points to get to strong, but a key variant is later found to be relatively frequent in NOMAD, do you downgrade the classification immediately, or do you wait for a formal reevaluation? So while ClinGen does have formal recuration timelines, it also takes community requests for curations or recurations, and will perform them earlier than required if something like this was brought to our attention. In general, for stronger definitive genes, there are numerous variants scored, so it would depend on the remaining data whether a curation would truly be downgraded or just the variant would be removed from scoring. And yes, there are certain disease areas that there really is one or two very common variants that make up the majority of cases. And so in that case, that might end up as a disputed curation, but we would take that back to the expert panel and have them review it as soon as we were made aware of that publication. Another recuration question: what strategies does ClinGen use to reassess genes that were originally classified as limited but have since accumulated new evidence, especially from regions with historically low publication output? So ClinGen has recuration guidelines based on our historical data. Limited genes have been shown that they don't necessarily change as often as other genes. Moderate genes, at least in our analysis, have shown that they move the most up most of the time. And so from our historical data, we have written a policy that moderate genes are the genes that should be reevaluated the most frequently. And so those are every 2 years, whereas limited are every 3 years. However, as I said, we also take community requests, and we're also exploring setting up automation and publication alerts to prioritize recuration, because we really would like to perform those recurations that would make a difference and change the classification instead of just doing them in order based on time. Okay, so now we have a handful of technical questions. So one of them is, why is it important to have a standardized framework for assessing gene disease associations, and what risks arise if laboratories use genes with weak or conflicting evidence in diagnostic panels? So when I give my talks, I really like to show this graph that now is a little bit old, where I went to the genetic testing registry, which is the public registry in the US that US clinical labs are required to upload all of their tests. And I searched for hypertrophic cardiomyopathy. and I pulled the number of genes in the hypertrophic cardiomyopathy gene panels from 14 different US clinical labs. And I was shocked to see the variety of gene numbers across these 14 different clinical labs. Some had under 20, whereas some had over 100. And so when you're a clinician ordering a test, which one of those do you choose? The one that's smaller or the one that's bigger? And I think in the past, the thought was it's a better deal for your money to order from a testing lab that has the most genes possible to be able to get an answer for your patient. But in general, we found that you need to have legitimate disease genes on your panel, or you're going to end up with a bunch of uncertain significance variants that you really have no idea what they're doing. And that's why we feel that standards for gene curation are very critical, because these labs that are just throwing a ton of genes on their panel just because they were published once to be associated with a condition are ending up with a bunch of VUSs in their panels. And the biggest risk is that labs will return more VUS if they include limited genes on their tests, or incorrectly classify variants as likely pathogenic or pathogenic in limited evidence genes. I can't tell you how many times I've acted as a paper reviewer in a gene discovery paper, and in a cohort analysis, authors will classify variants as likely pathogenic or pathogenic. And it turns out when I go to search the gene disease relationship in ClinGen, it's for limited genes. You cannot classify variants as pathogenic if they are in limited genes. Even for moderate genes, the highest level of classification you can get to is likely pathogenic. You need to be in a definitive or a strong gene to get to pathogenic variants. And that's the biggest risk when you don't systematically evaluate your gene disease relationships. Another technical question, what other methods can be used to um, design gene sequences apart from the ones mentioned in the articles provided? The ones that are not, some of those are not cost effective for African countries. So while the framework may give more points for technologies that sequence more genes than just the one being evaluated, for example, exome or genome or next generation sequencing panels, you are still able to score other technologies as long as they're identifying specific variant or variants in the gene that would implicate it in causing disease. So Sanger, PCR, any like restriction digests, if it really is going to show you a particular genotype, those are all fine. You just need to have a specific variant. You can't just say this gene itself as a whole, or there's a large copy number variant, like anything like that. You need to have a sequence variant in a gene to be able to score it within this framework for a gene disease relationship. It doesn't necessarily have to be in these more expensive, less available technologies. Another technical question is, how do differences in gene panel completeness, both in terms of technical coverage and the strength of evidence for gene disease association, directly affect a diagnostic test's clinical sensitivity and specificity? So this is very lab specific. So each lab, when they're doing their validation of a particular test and the content on their particular test, should calculate the sensitivity and the specificity of their test. So sensitivity, to have everyone on the same page, is the ability to detect true positives. Specificity is the ability to detect true negatives. And while this can be affected by the number of genes associated with a condition, you also need to consider the types of variants and the diagnostic yield of the genes when designing a test. And so at this point, there's no gold standard for the number of genes associated with a condition. And this can make it difficult because labs, while we're trying to standardize, evaluation of gene disease relationships, we have not curated all genes yet. And so labs often have had to curate these genes themselves. And now that we have the Gene Curation Coalition, we're at least comparing our curations to make sure that we're all on the same page. But there is no specific way that is determined for all labs to have to go about it. It's just they have to write about it in their standard operating procedures and their validation reports and be consistent with that and very clear in their methods and limitations. Okay, another technical question. How should a negative panel result be communicated to patients without false reassurance? Um, So I will mention again that I'm a laboratory geneticist, but clinicians could talk about sensitivity and specificity, like we just talked about for a particular test. They could also talk about diagnostic yield for a particular condition. There are certain conditions that really only a small portion of the diagnostic yield has been found to be genetic, and much of it is not discovered. That could be mentioned to the patient. And I think you need to have a different conversation for diagnostic tests versus screening panels. So for screening panels, clinicians could talk about residual risk. This often is discussed in carrier testing. So labs often have language like this in their methods and limitations for a test, and they often will say that a negative test reduces but does not eliminate the risk of a positive result. And as we've discussed before, it's different for diagnostic tests where your patient is affected with a condition, and treatment should still be geared towards their diagnosis and their symptoms in the absence of a genetic diagnosis. Of course, when you have a genetic diagnosis that can be added to that, you might have further management that you would enact at that point. But you should continue to treat them appropriately, even if they have a negative test, if they are affected. Okay, another technical question. Is the gene curation workflow workable among all nations, including Africans, where there are a lot of challenges like technology? Yes, in general, the framework encourages you to use publicly available data to perform gene curations. ClinGen ourselves, we do not generate any of the genetic data that is scored, although experts on the panels may bring some of their internal laboratory data that's been submitted to ClinVar that we can then use to score. But I would say that's probably more common in variant curations than in gene curations. But particularly for our framework, we make sure that things are publicly available before we score them so that everyone can check our work. The next question is on expert judgment. So what part does clinical expertise play in the final classification? So ClinGen semi-quantitative framework has flexibility inherently built into it. Each clinical validity strength has a range of points, you'll notice, and the ranges are not overlapping. So in my experience, experts are usually most critical for those genes that fall between the strengths. So for example, limited is 1 to 6 and moderate is 7 to 11. So if your gene scores at 6.5, you really need to have those experts evaluating the data to make sure that you've scored it correctly and to make the designation should the gene stay at limited and not be suggested to go on diagnostic testing panels, or should the gene score at moderate and be suggested to be put on diagnostic testing panels. I think that's the biggest critical distinction there is between limited and moderate across my expert panels. In general, the both my staff curators that are paid and volunteer curators, once they're trained extensively, are experts in the ClinGen framework itself, so how to use the gene curation framework. And they often have to teach the experts about how to use the framework. And how we would score things. The clinical experts are critical at evaluating the completeness and the reliability of the data that the curators find, because they're very familiar with clinical phenotypes, seminal experiments in the field, saying everyone is familiar with this animal model, which I might not know because I know the framework very well, but I don't know the disease area well. They've also potentially performed some of the functional assays themselves, so they can comment on experimental design and results, how reliable is the assay. And finally, they're also critical for weighing in on conflicting evidence, which is what we talked about before. What is the weight of that conflicting evidence? Should it bump an entire gene curation to dispute it, or should it just not be scored? Another question about expert judgment is how does reduced or age-dependent penetrance influence the interpretation of pathogenic variants in diagnostic gene sequencing panels? And what challenges does this pose for establishing gene disease clinical validity? So, when scoring variants in a gene disease relationship, we often rely on experts to comment on the phenotypic spectrum of a condition and the age dependent penetrance. And if that's not necessarily known or we're not sure about it, we will try to look for consistency across cases to determine that ourselves. And so, really, the curators will try to pull as many cases as possible, and oftentimes we'll put the phenotypes in a table and the ages of onset, for example, for us to look across all of the literature and say, what are the minimum common phenotypic features. What are the average ages of onset if this isn't readily known or available? And we also try to capture this and comment on it in our curations by capturing the age of onset or age of diagnosis, um, all of any ages that we can, to really get a sense of that. Okay, so another question about scoring. I think we're pretty close to the end of questions now. Is how does ClinGen incorporate evidence from unpublished data sets or internal cohorts from Africa, Asia, or Latin America where publication barriers exist? So ClinGen's only requirement is that the data are publicly available in some way so that others can check our work. They don't have to be published. So the simplest way to do this in these situations, and I've helped researchers from these countries to do this, is to encourage submission to ClinVar if possible, or other case level databases like Lovid, as we can score entries from both of those databases in our curations. And those entries in ClinVar and Lovid don't necessarily have to include all of the phenotypic information for a particular case. If a country or laboratory is concerned about sharing that, that could just be shared internally with the ClinGen expert panel, just for us to make a distinction of whether we should score something or not score something if it's consistent with the phenotypic spectrum. And so we have found that this is the best way for experts both in the US and internationally, if they have cases, but they cannot publish them, to submit them to ClinVar so that we can score them, or Lovid, any case level database. We also can score Decipher cases, but it has to be from the Decipher subset that's publicly available because we don't want it behind an access wall. So gene curation expert panels, how does ClinGen determine when a gene should move from the general working group to a dedicated expert panel? So for all expert panels, both gene curation and variant curation, each expert panel actually puts in an application. So if a group of experts in a particular disease area is interested in starting an expert panel, they have to fill out an application and come to ClinGen so that we just don't have a bunch of duplicative work, or very skewed representation on panels. They fill out an application to become an expert panel in ClinGen. And for each expert panel, we evaluate diversity of expertise and global representation. And so in this process, these groups also develop gene lists ahead of time so that we can make sure that they are not planning entirely duplicative work. So if the gene list that they've provided is entirely already curated by another expert panel, we would probably give that feedback and say, you need to consider a different gene list. Also, as groups curate their gene list, they may transfer a curation to another expert panel if it's more appropriate for them to curate. So when they're doing that, lumping and splitting that I talked about, like how many diseases are supposed to be asserted to be linked to a gene, they may decide that only one of those curations is relevant to them, and they might need to reach out to other expert panels within ClinGen and ask them if they would be willing to curate these other conditions. So one common example of this is I work with the syndromic disorders expert panel, which is really kind of a generalist expert panel of those disorders that don't necessarily have a major body system. They are really truly syndromic in nature and rare. And this group takes many curations from other groups because when they evaluate the curations, the other groups might realize that this is a rare syndrome and they just don't have the expertise for it. So they'll pass it along. Okay, and so with that, we, as usual, have some time for further questions. So I am going to stop sharing, and people can stick them in the chat or raise their hands. Um, I have a couple questions there in the chat. So in variant interpretation, how is penetrance typically determined? Is it mainly inferred from pedigrees and family studies, or are there standardized approaches to classify penetrance as high, medium, or low? That's a good question, and I think it's a difficult one. I would say people tend to be inconsistent with how they evaluate things. Um, ClinGen has a low penetrance working group that is trying to define low, moderate, and high penetrance. And I would say that I think the gold standard is, large kind of like case control studies or maybe like a really large analysis of um, families, cohorts. But as I'm sure you know that many conditions are rare, and so that often is not possible in many conditions. And also, as you may know, as we further evaluate biobanks, we, I think, are realizing that the penetrance estimates that we have are skewed in the high direction because those families with really high likelihood of genetic conditions that, have these variants segregating with disease, and have very severe phenotypes are the ones that come to our attention. So I think the penetrance is often lower than we realize on average for many conditions. And so there isn't necessarily always a great way to estimate this. And oftentimes it does involve just looking across small families or multiple studies and seeing what the age of onset is for conditions and how many individuals with potential variants in the genes are affected by a certain age. But there's definitely not necessarily a systematic way to do it. And when VSEPs are trying to come up with minor allele frequency cutoffs for their genes and their diseases, that's oftentimes the most difficult thing for them to estimate because the minor low frequency calculator does ask for estimates of penetrance. And I would say even for conditions that experts consider to be highly penetrant, they often will be conservative and say those are 80% penetrant for the reasons that I just mentioned, that I think we tend to overestimate penetrance because we're seeing those individuals that came to clinical attention because of their severity of condition. And then related to penetrance, I often see references to allele penetrance, gene penetrance, and disease penetrance. Are these distinct concepts in which terminology is considered most accurate in clinical genomics? So it's a great question. So there is potentially distinctions between those. So a variant might be low penetrance, and the rest of the variants in the gene actually might be high penetrance. So there might be a reduced penetrance for a particular allele. To go back to the hearing loss example, there is a particular mild hearing loss variant that case control studies have shown is actually pretty reduced penetrance. Gene penetrance is really like largely across the gene, and this would be, is your condition a high penetrance or a low penetrance? And that's kind of lumped together with disease penetrance, but disease penetrance is maybe related more to the disease area as a whole. So is your condition highly penetrant or not versus gene variants in your gene? Okay, let's go with the one person who has their hand raised. I think there is only one hand raised right now. Heba, you're muted if you're talking. Okay, maybe not there. So let's go back to the chat. I don't think I see other hands raised. One did just come, but I'll go back to the chat then. Um, there is an example, or there's a request to demonstrate real examples that exemplify gene curation and variant curation workflows. There are some videos on the ClinGen YouTube channel, so ClinGen resource, that would potentially take you through some real examples of gene curation and variant curation. So I'll refer you there, as we tried to go with more general overviews for this course. And then another question is if what we learned during this training course is taught or offered as training in universities and hospitals anywhere in the world. I would say yes and no. I think that's why I often am asked to give workshops throughout the world, is because people are realizing how important learning this information is, and they don't always have access to materials to be teaching these courses. So I think we've tried to do our best and train the trainer workshops. So I've been invited to give workshops in Argentina, in Brazil, in Italy, in Qatar. This workshop, I'll be giving another workshop in South Africa. So we really do try to travel or work with groups who are willing to learn. I would say that in the US, it has become a little bit more widespread, that it's a bit more of a residency requirement. So when people are in medical school, they're starting to teach genetics more and more. But some of this really in-depth information that we're talking about, so curations, is really limited to genetics fellowships. So really those who plan to specialize in genetics. And I agree that at least some basic level of information can be really critical because there are plenty of non-genetics physicians ordering testing, or people come with genetic testing reports if they did direct to consumer testing with their primary care physician or someone else, that this is really important to be learning. So we're trying our best to work with everyone and provide resources to help to teach things like this. Okay, let's go with some hands here. So Heba, are you back? Are you able to speak now?
I hope I am. Can you hear me?
Yes, I can hear you now.
Oh,
Yes,
perfect. It was a technical problem. I'm sorry about that.
no
Um,
worries.
Thanks. There was a note about the presence of the homologous pseudogene juxta to the gene of interest, and how this can make it difficult to interpret the variant found, or even the annotation of the sequencing data. And the example given was in Lynch syndrome with PMS2. And so I just want to understand how this happened, and particularly when the variant is in the sort of the terminal end of the coding sequence in the untranslated region, and still it is sort of scored as a likely pathogenic variant just because it's in in the hotspot area. Uh, I really don't know how to interpret this really, and score it as a pathogenic. I know this is not this session is not very much related to variant interpretation, but it was mentioned in the sort of the article about gene disease correlation.
Yeah,
So
it's a good question. So I will say the difficulty with pseudogenes is sometimes placing a variant in a gene versus a pseudogene, of course, because sometimes you classify a variant and it would be a likely pathogenic or a pathogenic variant if it was in the real gene, because it's maybe a loss of function or a variant that would be damaging. And so the difficulty therein lies in distinguishing if it's in the real gene or the pseudogene. And I think the critical point there is to evaluate what type of testing was done. So as someone who's in a clinical lab, we have very specific additional pieces of testing that we do in highly homologous regions like long range PCR, for example, digital droplet PCR, to really try to be sure that those variants that would be reportable, would be likely pathogenic or pathogenic, are actually in the disease causing gene and not the pseudogene. Because yes, you can easily get a variant to likely pathogenic, and that's the problem. If these variants were not considered likely pathogenic or pathogenic, you wouldn't have to worry about making sure if it was in the gene or the pseudogene. So I think the classification of the variant isn't necessarily the issue. It's just making sure that it's the real variant versus the pseudogene variant. Because if it's a loss of function variant, it would be damaging and it would be likely pathogenic or pathogenic. It's just really making sure, either in your patient or in cases you're scoring in the literature, that the appropriate testing was done to be sure of that.
So basically, it has to be through validating this variant through an additional testing, like the long range
Yes.
digital,
Hmm.
and this
Yeah.
can tell whether it's in the true gene or the pseudogene by sort of the
Yes.
primary.
Yeah.
Okay, perfect.
Hmm.
Thank you very much. Cheers.
Of course. Yeah. So I got a question in the chat about what about my Nigeria for workshops. And so I will say that ClinGen has worked closely with H3 Africa and has come to Africa a handful of times actually to give workshops there. But we are always willing to work with groups for workshops. So if anyone has any requests or thoughts about workshops for particular locations, whether they be in person or remote, you can definitely just email me or email ClinGen at clinicalgenome.org, Oregon, we can try to accommodate that, with the caveat being that most of the time our funding is pretty scarce or strict. And so if it does require travel, we may need to do a virtual workshop if that can't necessarily be funded or provided, because we know that everyone has a little bit of tight funding at this point, but we're always willing to help in whatever way we can, like I'm doing with this virtual workshop. Okay, I think there is one hand raised. Is there someone who wanted to unmute with a hand raised? I can't hear you if you're trying to talk. You are still on mute. Okay, we'll go to the chat then. Um, Actually, I think I've answered all the questions in the chat that are related to gene curation or this talk. So are there any other questions that anyone wanted to ask?
I
Okay.
think Omar from Uganda, he's trying to speak, but we don't hear you.
No, we don't.
Oh, yeah.
Can you hear me now?
Ah, yes.
Yes, yes.
Okay, thank you so much, Dr. Marina, for the wonderful presentation. Very clear, very elaborate. We really appreciate so much. I have one question. We have had a lot of sequencing in African projects, H3 Africa being one of them. So I would wonder whether so far we have come up with any data from African genome that we could use as like a reference. I mean, understand right now every samples are being called against the global human genome project, the one that was done originally. So I would want to know if we have any like an African reference where maybe the samples from Africa could be, checked against the African genome. Thank you so much.
Yeah, I mean, I think I encourage all groups that are generating like very large reference data sets to use their reference data sets, because I think that's the best comparison for, patients with rare disease in your country. And I will say that I think that at least internationally, people have, with telomere to telomere efforts, for example, or the Pangenome Project, where they're trying to incorporate as many reference genomes globally as possible into one large reference genome, is trying to do a better job of representation of globally, of reference genomes, because those original reference genome projects were really like five guys in New York or something. Like it was not, even representative of the United States, let alone of the world at large. So I would encourage researchers to really try to use their own internal reference data. I will also say that Nomad has really started to embark on a project that is a federated Nomad, because in the past, Nomad, in order to ingest data sets, has had to actually have access to the data so that the same variant calling and bioinformatic pipelines could be applied across all of the data set, which really has posed challenges internationally with data sharing, and rightfully so, because you shouldn't be handing over your data to all be held in one place. And so the most recent work is, their goal is to create a federated Nomad where there is like wheels or a spoke wheel model where each country holds on to their data, but everyone who comes can query all of the countries with their data. And so I think they are trying to get many of these genome projects across the world to be part of this federated Nomad, so we can really truly have global representation. And so I think that's kind of some of the efforts that we're working on to really expand these reference data sets. But definitely you should work with your own reference data set when you can, because it is a very good reference for your local populations. I
Here is
think,
Osman
yep.
Muhammad from Nigeria. Osman. Al-Hassan also wrote in chat from Senegal. Okay.
Oh, yeah.
Osman. Okay.
Maybe is a technical problem, but there is a question in the chat of how you classify variants in non-coding regions. So there has been some specific work that has been done. Let me see if I can pull it up. And this question actually came up last time on classifying non-coding variants. Nikki Whiffin's lab in the UK has been working on this. I want to see if there's guidance on the website in ClinGen for this to be able to put in the chat for you. Let's see. Yes. So here is the link to the guidance that ClinGen recommends for non-coding. Actually, it's not letting me. Okay, no, it's not letting me type in the chat. Let me try again. Okay, I'm gonna screen share it so that you can see it and then maybe like grab the PubMed ID if that's helpful for you, because I can't seem to stick it in the chat. This is the PubMed ID here. This is recommendations for clinical interpretation of variants found in non-coding regions of the genome. So feel free to grab a screenshot of that and take a look at this. Okay. So another question about reference genomes, which reference genomes are currently used in clinical settings? Is it HD38 or T to T? It's a great question. I would say, unfortunately, in most cases, it's probably HD38. I know there are some labs that are even on GRCH 37 at this point, and it's because clinical pipelines, once they are validated, are relatively inflexible a lot of the time, that you really have to move a reference revalidate your entire pipeline. But I would say that many people are trying to move to T, particularly for certain types of variants, like mitochondrial variants or other variants that are just better represented there. So our lab is definitely doing some exploration with the T reference genome, but we are not clinically reporting on it right now. We're on GRCH 38. Okay, so I think we are at time. Great questions and great participation again, everyone. I look forward to the last email that I'll be sending you for week 4 materials on actionability curation and that quiz. As I said, everyone, please participate and take that quiz. I'll reiterate that in the email that I send along, and I hope you all have a wonderful weekend. Thank you.
Thank you so much. Have a good day.
Yeah, bye.
Thank you.
Bye-bye.